Hi, I'm Aaradhya,
And I'm Sajid.
and our topic is the functions of CRISPR Cas9.
In 2012, scientists Jennifer Doudna, Emmanuel Charpentier, and Feng Zhang developed a gene modification process that could target specific genes with an extreme amount of precision and only modify that specific gene. That process was named CRISPR-Cas9. Although it would be years till this process was implemented on humans, scientists noticed that theoretically this could be used to cure human sicknesses. In 2020, the creators of CRISPR-Cas9 received a Nobel Prize, recognizing their efforts for pioneering one of the only methods of genome editing that could theoretically be used on humans. Now, I will explain how CRISPR-Cas9 works and how it's so precise. CRISPR-Cas9 is a process which can be used to modify and isolate different genomes and genes, simply to find specific bits of DNA inside a cell and then alter that DNA. This process can be split into two, CRISPR and Cas9. But first, let's go through what this means. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, describing a region of DNA made up of short repeated sequences with so-called spaces sandwiched between each repeat. Cas9 is an enzyme used to cleave through particular segments of DNA, essentially acting as scissors. Simply, CRISPR is a guiding device which guides Cas9, kind of like molecular scissors, to target a section of DNA. Together, they work to modify that gene, acting as kind of a genome missile.
About how CRISPR-Cas9 was developed. This gene modification method was derived from a naturally occurring gene editing process in bacteria. Within this process, bacteria captures segments of DNA from invading viruses and uses them to construct DNA segments known as CRISPR arrays. This allows the bacteria to somewhat remember the DNA of this particular virus, and the next time it attacks, the bacteria produce specific targeted RNA from the CRISPR array to target the virus and use an enzyme similar to Cas9 to cleave the DNA, disabling the virus from functioning. This process works similarly in a laboratory. Researchers produce a small piece of guide RNA which attaches to the target region of a particular DNA sequence. The RNA is used to identify the DNA sequence, and then the Cas9 enzyme cuts the DNA at the particular region. Once the DNA is cut, researchers use the cell's own DNA repair machinery to turn specific genes on or off, or replace an existing DNA segment with the custom DNA segments. Normally, with other methods of gene editing, an enzyme such as ligase is used. This is because when the DNA is cleaved, the phosphate sugar backbone is often left exposed. Ligase acts as somewhat glue and helps repair these separations. CRISPR-Cas9 uses the cell's natural DNA repair machinery to form bonds between the edited DNA and the existing DNA. Cells cannot survive for long with cut DNA, and this immediately sets steps to patch up the DNA.
When compared to other forms of genome modification, CRISPR-Cas9 only cuts gene at one specific point. This is due to the fact that guide RNA can be made corresponding to a specific gene locus, ensuring only one gene is getting cut out. This process can modify genes in a variety of ways, turning specific genes on and off, ensuring certain genes are not present, and even replacing certain gene fragments with custom gene fragments to modify certain alleles. This can theoretically be used to change factors like eye color or height. This process has generated a lot of excitement in the scientific community due to the highly precise method used for CRISPR-Cas9 and it being faster, cheaper, and more accurate than other research methods. Our article, titled The CRISPR Baby Scandal, What's Next for Human Gene Editing, an article present in the Nature Medical magazine written by David Saranowski in 2019, dives into the ethical implications of CRISPR-Cas9, using a practical example to foreshadow the extent of possibilities CRISPR procures. This article revolves around the controversial birth of two baby girls born with edited genomes, made possible by the work of He Jiankui, with Saranowski describing Jiankui's work as leaping capriciously into an era in which science could rewrite the gene pool of future generations. Saranowski explains his view on this experiment, alongside highlighting the future of ethical violations that may be accredited to CRISPR-Cas9. We chose this article as we concurred it presented an interesting case of one of the few practical large-scale experiments involving CRISPR-Cas9, as well as diving into this topic to view the long-term reality this discovery brings.
CRISPR-Cas9 is important because it allows scientists to edit the genes of virtually any organism, regardless of its species. It is simpler, cheaper, and more precise than previously developed gene modification techniques, whilst having a wide range of real-life applications. CRISPR can be used to cure various genetic diseases and modify crops and plants to make them easier to grow. CRISPR-Cas9 is relevant to all demographics. However, if it is viewed from a medical standpoint, it has the potential to aid several sicknesses present in developing countries. Children under the age of 5 suffer from significant mortality risks around the world, specifically in developing countries. CRISPR-based strategies could reduce the morbidity from these diseases to a high extent. This could be done by gene editing mosquitoes to prevent malaria, which could cause a significant population of people to be protected. In Africa alone, approximately 285,000 children under 5 years old die from malaria in 2016, and nearly 500,000 individuals globally die from malaria every year.
CRISPR-Cas9 has helped crop farmers achieve remarkable results. Within the last decade, this novel method of molecular editing has become the most used to make plants more effective. CRISPR-Cas9 has accelerated the crop breeding process by its precision in specific gene editing. Some of the ways these crops are being modified is by making them more physically appealing. For example, making strawberries larger and more vibrant, which increases consumer purchase rates. Additionally, having larger fruits means that more crops measured in weight are yielded with the same amount of labor, making the farming process more economic. CRISPR-Cas9 can also insert certain genes into plants to make them resistant to a variety of abiotic and biotic factors, such as drought, pests, or even pesticide itself. An interesting example is having plants resistant to pesticides, as within this process, pesticides can be sprayed directly on plants. Farmers spend about $10,000 each year protecting their crops from pests, and the development of this technology can decrease that amount by eliminating animals from affecting crop growth negatively, and once again increasing crop yield.
Additionally, the technologies propagated by CRISPR are also relevant to all of us. This is shown by using CRISPR to develop certain booster COVID vaccines. An article by Alex Marston describes the progression of a CRISPR-based DNA vaccine enhanced for COVID-19 that would radically reduce the timeline to develop vaccines against current and future viral threats. The advantage of this type of CRISPR-based vaccine is that the immune responses will be improved from a singular dose. While this is practical for citizens, as they do not have to take as many vaccine shots, it is also economical, as a greater population can be immunized with one vaccine stock. The main implications of CRISPR-Cas9 are the infinite unknowns that come with gene editing. Our main article accentuates this fact by discussing the moral implications that came with He Zhuangai's experiment, the man who engineered the first gene-edited twin babies with a resistance to HIV. In June 2016, He Zhuangkai began a project which used CRISPR to edit a human embryo's genes. His main goal with this project was to edit the genes of a baby whose father was positive with HIV, and in March 2017, he started trials with couples with a HIV-positive father. He claims to have prevented a specific protein which encodes for HIV to enter the cell by disabling a gene called CCR5. This is a mutation which helps protect them from HIV infection, as already in 10% of Europeans. He developed these mutations on a human embryo and produced babies. These mutations all had to be done on a human embryo, as it has drastically less cells than an adult human. This would make it easier and more effective.
However, while doing this, he may have inadvertently caused mutations in other parts of the genome. This is due to the fact that CRISPR at this stage is just too recent, and that method was not optimized for human use as of yet. This could have an unpredictable number of health consequences. Not only this, but CCR5 is also responsible for the body fighting off many other infections, such as the West Nile virus. This could leave the babies vulnerable to many other diseases. It was then widely condemned as a dangerous and irresponsible experiment, and he was later to serve 3 years in prison for illegal medicinal practices. Another study found that CRISPR-Cas9 can cause devastating defects in the cell of an embryo, and cause them to discard large portions of their genetic material. The CRISPR-Cas9 technology was used on specific cells, which can cause hereditary blindness, and it appeared to destroy it in approximately half the specimens of the experiment.
Furthermore, another ethical problem to consider is the application of CRISPR-Cas9. The original purpose for CRISPR-Cas9 is to edit the genome to mutate genes such as CCR5 for HIV and CFTR for cystic fibrosis, and prevent the disease from occurring. However, many bioethicists are concerned that once this practice stabilizes, it will then be used for commercial purposes to enhance children rather than for strictly medical use. In essence, you could change things such as a baby's height to create designer babies. This could purposely make some babies more athletic or superior in many physical ways. This crosses an ethical line as it essentially changes parts of a baby unnecessarily, which alters the baby's identity, questioning human nature as a whole.
CRISPR-Cas9 is a comparatively cheap solution to gene editing. Gene editing has been prevalent in the past but has never revolutionized in this way. An article from Vox states that in the past it might have cost thousands of dollars in months to alter a gene, but with CRISPR it could take $75 in only a few hours. Due to CRISPR Cas9 being such a recent discovery and the lack of practical application at this stage, we haven't seen much of an economic impact. But considering the development of other GMOs, it's been projected to produce great economic growth, with reports stating that crop biotechnology has drastically reduced agriculture's environmental impact and stimulated great economic growth in Australia.
Now, we move on to our 3 questions about the topic. In researching this topic, to a high extent, we questioned the data we found at several places. One of our main questions was relating to the application of CRISPR Cas9. If this technology really has the potential to cure genetic diseases, why is it not being implemented? In 2019, over 5.2 million children under the age of 5 died from preventable diseases. If something can be done, why is it not?
Additionally, we wondered about the future of CRISPR Cas9 and if it's inevitable that it will turn into a commercial business. Finally, we questioned how this process was developed and what steps were taken by the team of scientists prior to publishing their findings about this process. Thank you.